Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we're going to talk about genetic exchange in bacteria. Bacteria reproduce asexually by binary fission, but genetic diversity is maintained through three major mechanisms of horizontal gene transfer: transformation, conjugation, and transduction. These processes enable bacteria to adapt quickly to environmental pressures such as antibiotics, leading to rapid evolution and the spread of resistance genes. Let's start with transformation. Transformation involves the uptake of naked foreign DNA from the environment into the bacterial cell. This DNA, often released by lysed bacteria, is integrated into the host genome. Many gram-negative rods and some gram-positive bacteria are capable of natural transformation. This process enhances genetic diversity and allows acquisition of new traits, including antibiotic resistance. Conjugation is the bacterial equivalent of mating, and it involves direct cell-to-cell contact. This is a unidirectional transfer of genetic material and is mediated by a plasmid called the fertility factor. It is annotated as F. Bacteria with this plasmid are termed F positive, and those without it are termed F negative. So what happens is a donor cell, F positive, transfers genetic material to a recipient cell, F negative, via a sex pilus. Once transferred, the recipient becomes F positive and can initiate further conjugation. A quick note is that the sex pilus that they are transferring genetic material through is also called the conjugation bridge. Moving on, if the F factor becomes integrated into the bacterial chromosome, the bacterium is termed an HFR cell. HFR stands for high frequency of recombination. During conjugation between an HFR cell and an F negative cell, chromosomal genes adjacent to the F factor can be transferred, though the entire chromosome is rarely transferred due to time limitations. This process is often used in bacterial gene mapping. Let's quickly talk about conjugation mapping. HFR bacteria provide a mechanism of mapping the bacterial genome. By allowing HFR cells to conjugate in the lab and stopping the conjugation process after different time intervals, researchers can figure out the order of genes on the bacterial chromosome by analyzing recipient cells to see what genes were transferred. So for example, you have two strains of E. coli. One is a normal HFR bacterium and the other is F negative and an auxotroph for arginine, leucine, and histidine. So it's F minus and then it's arginine minus, leucine minus, and histidine minus, which means it does not have these. So now you allow conjugation to begin and then you stop it after 2 minutes. You find that all the recipients now are still F minus, arginine minus, leucine minus, and histidine positive. Then you take another bunch of bacteria and allow conjugation to proceed for 5 minutes. Now all the recipients are F minus, arginine positive, leucine minus, histidine positive. You do the experiment a third and final time, allowing 8 minutes of conjugation, and find the recipients are F minus, arginine positive, leucine positive, and histidine positive. So from here, what we see is that the ability to make histidine was transferred in a short time. After a slightly longer time, the ability to make both histidine and arginine was transferred. And after a slightly longer time, the ability to make histidine, arginine, and leucine was transferred over. So in conclusion, we know from this that the map must be histidine, arginine, leucine, and then the plasmid integration site. Now let's move on to transduction. Transduction is a gene transfer mediated by bacteriophages. These are viruses that infect bacteria. During the assembly of new phages, host DNA may accidentally be packaged into a viral capsid. When this phage infects another bacterium, it can inject host DNA, which may integrate into the recipient genome. This mechanism allows for precise gene transfer and plays a major role in bacterial evolution. And finally, we have transposons. Transposons are mobile genetic elements capable of moving within and between genomes. They can disrupt gene function by inserting into coding regions and may carry antibiotic resistance genes. Transposons are found in both prokaryotes and eukaryotes and contribute to genome plasticity and evolution. That's it for today. Next time we will go over viruses.